How do you write a sentence? Maybe you start with a rough draft. You check for typos and fix mistakes. Maybe you even substitute words for better ones. Now what if I told you that we could do the same thing, but with genes? What if we could erase genetic mistakes and fix the mutations that cause diseases? What if we could make our immune systems more effective and improve crops? Well, actually, we can. For the past few decades, scientists have been learning how to write in the language of DNA. They've developed many tools to help them work on this molecular level, and improvements are developed every year. Two of the most exciting are CRISPR-Cas9 and base editors. CRISPR-Cas9 is one of the most popular of many gene editing techniques that use double-stranded DNA breaks. But what makes it so special is that it's cheap, accurate, and easy to use. The Cas9 protein by itself is aimless, but when guided by RNA to specific locations in DNA sequences, it creates double-stranded DNA cuts. When a cell's repair mechanism sticks these cuts, random insertions or deletions are usually included, which results in the targeted gene becoming ineffective. This means that Cas9 is very, very good at turning off genes, and scientists can use this effect to modify organisms. Cas9 can also be used to add new DNA sequences if there's a template, but this process is in constant competition with normal repair, it's error-prone, and can only occur when cells are at a certain stage of the cell cycle. And although improvements to this process can be made with more chemicals, additional complications and unpredictable errors aren't exactly ideal when working with DNA. So Cas9 is like these garden shears. They're great at large-scale work, cutting genes and turning them off, but for smaller modifications, we need something else. And that's where base editing comes in. To understand base editing, we'll first need to know a little bit about DNA structure. DNA's nitrogenous bases are like pairs of shoes. A pair of shoes with matching color, but need to have opposite shapes. Similarly, only A and T and G and C can be matched with each other in DNA. Now back to the base editors. They come in two parts, a deaminase and a partially deactivated Cas9 called Cas9 nickase. Like normal Cas9, the nickase can be guided by RNA to specific locations along a piece of DNA. However, the nickase does not create the double-stranded break that normal Cas9 does. Instead, the deaminase will chemically convert a targeted base into another by adding a water molecule and removing an amino group. Now the two bases don't match, so the nickase will nick the opposite strand, which tricks the cell's repair proteins into swapping out the old base to match the converted base. And that's base editing. Seems pretty simple, right? Well, that's actually just the basic process. Each of the 6 transformations of base pairs requires slightly different proteins and steps to stabilize the transformation. As of right now, only 4 have been developed. But those 4 transformations alone are capable of correcting almost 25,000 genetic diseases with their single base transformations. In the near future, base editing could eradicate genetic conditions like cystic fibrosis and sickle cell anemia. Even non-genetic diseases might be addressed. With base editing, we can permanently reduce cholesterol production and engineer anti-tumor T cells. Base editing has even been used to improve agriculture by increasing production of rice, wheat, corn, and tomatoes. Now that humanity is equipped with both CRISPR-Cas9 and base editors, who knows what innovations and discoveries will happen next? Maybe together, these tools and new ones can lead us to a brighter, healthier future.